So welcome back, everybody, to the second part of the morning session. Our first speaker is Noa from the ANU. It's a pleasure to welcome here and to have you. I think it's your first time, nevertheless, in the Analysis and PDE seminar in Australia. And she will speak about bifurcation, competitive evolution of network morphologies in the functionalized Cahn-Hilliard equation. Please start.
Thank you, Daniel. I'm interested in lipids. Lipids are what makes cells membranes. They have hydrophilic heads and hydrophobic tails. It is known from experiments that amphiphilic materials can form different types of structures, such as the bilayer, which is a sphere. It separates two regions of phase A by a thin layer of phase B, and filaments, which look like spaghetti worms, and they have their tails pointing in. We're interested in a mathematical model which can describe the network formation of such materials, as well as the evolution, as well as the behavior of each of the different structures. Today, I will introduce the functionalized Cahn-Hilliard free energy as our mathematical model. It is simple enough to analyze, and it can capture the behavior of amphiphilic materials. Our motivation comes from experiments. First, let's look at Disher and Eisenberg experiment. Here, they took diblock lipids and changed the volume fraction of the solvent.
I think your screen is, sorry to interrupt, but I think your screen is
frozen?
Yes, or at least not shows what you want to say. Yeah.
Let's try again. Do you see?
Now it's good.
It's changed. It's changing now.
Now
it's good. So yeah. Let's try again. And when I do this, does it work now?
No.
Oh, I think it's the full screen problem.
The full screen. Okay, I'll skip the full screen. Okay, sorry. So in case you missed it, that's the lipids that have
what a beautiful slide. What a beautiful slide that we didn't see.
Hydrophobic tails. These are the different structures. We have filaments, which are like spaghetti worms, and we have the bilayers. Okay, so in this slide, we're looking at Disher's and Eisenberg's experiment. In this experiment, they took diblock lipids and changed the volume fraction of the solvent. This is a water dioxide blend. When it's pure dioxide, then the lipids are hydrophilic. But as they add more and more water to the system, the lipids go through bifurcation. They change morphology. On the left, you can see codimension 3. When they add more water in the middle, they change to codimension 2. And as they add more and more water, they go all the way to codimension 1. Another experiment was done by Jane and Bates. They took 2 sets of diblock copolymers, and they changed the percentage of lipids in the background. Here, the y-axis is the length of the diblocks, and the x-axis is the percentage of lipids in the background. And as you can see, if we look at both chains for n equals 45 and for n equals 170, each set go through a sequence of bifurcation. They change from B plus C, all the way to S. However, if you look at the long chain, it is also interrupted by a Y-junction filament dominant pearl cylinders. So I'm going to show you the functionalized Cahn-Hilliard free energy to describe this kind of bifurcation. I cannot talk about functionalized Cahn-Hilliard free energy without explaining a little bit or reviewing the Cahn-Hilliard free energy. So Cahn and Hilliard wanted to describe the spinodal decomposition of binary mixtures. Here we have omega in R3 is a bounded domain. U is the volume fraction of one of the species. And we take a free energy, which is modeled by a function of U, weakly perturbed by its spatial gradients. If we Taylor expand this free energy and we integrate by parts the B term, then we get the classical Cahn-Hilliard free energy. Now here, if you think of water and oil, then the first term is trying to mix the two, that's the entropic effect, and the second term is the mixture potential. The interface is the thin layer between the water and oil, and the width of the interface is controlled by epsilon. And here we take W to be a tilted double well potential with two non-degenerate minima at plus minus B. It was suggested that for amphiphilic materials, we'll take the next order term in the Taylor expansion. If we do that, we see that we get a quadratic polynomial in Laplacian U. Again, we integrate by parts and then we complete the square and we get this nice form of F tilde. And F tilde is a very special form because if P tilde is equal to 0, then all we have is the square of the first variation of Cahn-Hilliard, which means that global minimizers are precisely critical points of Cahn-Hilliard. Okay, to make the analysis possible, we choose a specific form for P tilde. In this case, we have eta1 and eta2, which are the functionalization constants. The first term with grad U rewards surface areas, while the last term is the capillary pressure. Now, it's important to distinguish between the Cahn-Hilliard and the functionalized Cahn-Hilliard free energy. So the Cahn-Hilliard free energy comes to describe hydrophobic materials, while the functionalized Cahn-Hilliard free energy describes amphiphilic materials. This amphilicity derives the functionalized Cahn-Hilliard to operate on a different length scale. So if we look at the picture on the left, we have what looks like an early stage of spinodal decomposition. As we zoom in on B, you can see the walls between the interfaces. And if we keep zooming all the way to D, you can see the network formation within these walls. That's where the functionalized Cahn-Hilliard free energy lives. So basically, we had to zoom in 500 folds to move from Cahn-Hilliard to functionalized Cahn-Hilliard. More importantly, we moved from an electroneutral system to a non-electro neutral settings. And that's the difference between the two. Okay, to analyze the functionalized Cahn-Hilliard, we need a suitable coordinate system. So we introduced the Whiskered coordinate system. We assume that we have a smooth codimension 1 interface gamma, which separates omega into omega plus and omega minus. We assume that it's far from self-intersection. And assuming that it is far from self-intersection, we can find an open neighborhood around this interface such that each point X I can define in terms of X and S. I'm sorry, each point X I can define in terms of Z and S. So here gamma of S is the parameterization of gamma. Z is the scale distance from the interface and N is the outer normal. Using these Z and N coordinates, we can rewrite the Laplacian in a simpler form where the Z derivatives are separated from the S derivative. And we get this form here, age restricted to gamma is the mean curvature, and Laplacian G restricted to gamma is the Laplace-Beltrami operator. We can do the same for codimension 2 structure in R3. We get a different but similar expression for the Laplacian. Since minimizers of functionalized Cahn-Hilliard free energy are at leading order critical points of Cahn-Hilliard, we're looking at the unconstrained Euler-Lagrange equation. Inside the ridge, inside this small neighborhood around the interface, I can change coordinates to my Whiskered coordinate system, and I introduce the Bilayer profile, which is the homoclinic solution. And that's for codimension 1, that's for the bilayer. For codimension 2, for filaments, we consider radially symmetric solutions and we get the filament profile. Now, For those of you who know Cahn-Hilliard, it is important to understand that the bilayer solution is not the singular solution. The first thing, obviously, is that they separate different things. So a single layer solution separates water and oil. The bilayer solution is like a soap bubble or a beach ball. It separates two regions of phase A by a thin layer of phase B. Both structures are sensitive to low frequency perturbation, which will lead to geometric motion. However, the bilayer is also sensitive to purling. And the bilayer can use to model cell membranes. We can talk about opening a hole in the interface, and that just doesn't make sense for the single layer. Okay, let's make sure that the single layer and the bilayer are possible minimals for the functionalized Cahn-Hilliard free energy. So we introduced the linear operator LB or LS, which is on the real line. And if we take the Z derivative, we see that the Z derivative of U sub B is in the kernel of LB. And using Sturm-Liouville, we can say that this is the first excited state of LB. We take an expanded operator that will be the second variation of Cahn-Hilliard. Now for V supported inside the ridge, this script L operator takes this form. We can rewrite it using our new coordinate system. And it was shown that for this operator, the form of the eigenfunctions corresponding to small eigenvalues take this form, it's psi j times theta n, where psi j are the eigenfunctions of the linear operator, and theta n are the eigenfunctions of the Laplace-Beltrami. We can look at the corresponding eigenvalues, and they take this form, and we can see if we plot them that the single layer is indeed a minimizer for the Cahn-Hilliard free energy, but the bilayer is a saddle point for the Cahn-Hilliard free energy. The single layer is sensitive to low frequency perturbation. And lucky for us, the bilayer is not a minima for the Cahn-Hilliard, but we're looking at the functionalized Cahn-Hilliard. So let's just make sure that it is a local minima for the functionalized Cahn-Hilliard free energy. So if U is a critical point of Cahn-Hilliard, then the second variation of the functionalized Cahn-Hilliard free energy is actually at leading order, the squared of the second variation of Cahn-Hilliard, which means that all the spectrum of the bilayer is now positive. So at leading order, we can say that the bilayer is a possible minimizer for the functionalized Cahn-Hilliard free energy. It is, however, sensitive to high frequency perturbation, which will lead to purling, as well as low frequency perturbation, which will lead to geometric motion. Okay, let's start talking about the formal analysis of the geometric evolution. And the simplest mass-preserving gradient flow for the functionalized Cahn-Hilliard free energy is the H-1 gradient flow, which leads us to consider the equation U sub t equals to Laplacian mu, where mu is the chemical potential. Now, I forgot to mention, but let's do it now, that for this part of the lecture, I will assume that everything is Perlin stable. So I'm only dealing with the geometric motion at this point. We're going to start by looking at two separate systems. I'm going to take a system that has only the bilayers in it and a system that has only the filaments in it. So for the pure bilayer system, we get an expansion of this form where U sub B is the homoclinic solution that we introduced before, and it's slightly, it is perturbed by a small perturbation that depends on the chemical potential. We find that the normal velocity is coupled to the chemical potential, and the chemical potential is determined by the total mass of the system. And the evolutional interfacial area the evolution of the interfacial area is the integral of the normal velocity times the curvature, the mean curvature. So that was for Bilayers. We can do the same for filaments. And as you can see, we're getting something very similar. We have the formal expansion, which again depends on the chemical potential. The normal velocity is coupled to the chemical potential. And the expressions are really similar. So we see that the H-1 gradient flow drives pure bilayers and pure filaments by a quench mean curvature flow. Now here, the sign of V is very important. If V is negative, then this turns into a backwards heat equation in the curvatures, which means that the system will no longer be well posed. But assuming that everything is well posed and that V is positive, then we can see that mu1, the chemical potential, will decay into constant, and the constant will be different for each system. Okay, so now we can take, again, assuming everything is Perlin stable, we can take a system that has both the bilayer and the filaments in it. And so again, to remind everyone, the bilayers are the beach balls and the filaments are the spaghetti worms. And we're going to see that the two structures are going to compete through the chemical potential. The evolution of the interfacial surface area is coupled through the chemical potential, which is determined by the total mass of the system. In this case, when we have a mixed system, then the total mass of the system is composed of the mass in the background, the mass of the bilayer, and the mass of the filaments. Since B and F take different values, we know that the two structures will go to different equilibria, and we do not expect to support the coexistence of the two structures. So, so let's see some okay, so fixing W and eta 1, we can change eta 2 and find the values of F and B. So the blue line is the line for the bilayers, and the red line is the line for the filaments. Both structures are stable below their line and unstable above. So we get a region where both will shrink, a region where both will grow. And then we have the two gray regions where one will shrink and the other will grow. Now these gray areas are invariant, and as we see in these settings, the functionalized Cahn-Hilliard does not support in general the coexistence of the two structures. So here's a numerical simulation. We started with a single bilayer and two rings, which are two filaments. And we let the system run, and we can see that pretty quickly the bilayer will grow at the expense of the filaments. Okay, so now we need to investigate the Perling modes. And we look at the second variation of the functionalized Cahn-Hilliard, and we consider the eigenvalue problem. Now we know that for constant curvature, for constant curvature codimension 1 structure, everything will be Perlin stable as long as the charge level is lower than the lipid level. Okay, that was shown by Arian. And we want to use that, but I'm not looking at constant curvature. I'm looking at generic interfaces. So we took L, we decomposed it into this 2x2 block. Here, pi is the projection onto the space of small eigenfunctions, and pi tilde is the complementary projection. We know that pi tilde, L pi tilde, has a lower bound independent of epsilon. We show that the off-diagonal terms are uniformly bounded as an operators from L2 to L2. which means that all we need to study is this operator, finite operator, pi L pi. Well, it is finite, but it is big. We know from Wales' asymptotic formula that pi L tilde pi is big, and the eigenvalues are coupled through the non-constant curvatures. Luckily for us, if kappa, if the curvatures are bounded in the W to infinity norm, then we can write pi L pi as a diagonal matrix plus a small correction, but this A again is uniformly bounded as an operator from L2 to L2. So we get that at leading order, we have this diagonal matrix, and this diagonal matrix is exactly the same as we get for the constant curvature structure. Okay, so we showed excuse me, that for both the bilayer and the filaments, everything will be Perlin stable as long as the charge density is lower than the lipid level. And now we can talk about the full system. And I was super running. Now we can talk of the full system. So we can consider both the bilayers and the spheres. And we can consider both geometric motion and the purling. We can see that the purling will occur on a much faster time scale than the meandering. And yeah, and I had one more thing. And if we fix the double well potential and eta 1, we can graph the purling lines exactly like we did before for the meandering lines. Okay, so we obtained a full bifurcation analysis of the system. And I was going way too fast, so it means that we'll have a lot of time to look at the video. So let's look at some numerical analysis. Oh, I'm sorry, numerical simulation. So here, well, I'll stop here and I'll say what we're looking at. We're looking at the bilayer. We start with a ring, and it has way too much lipids in the background. So quickly enough, so at the initial position, it is both Perlin unstable and meandering unstable. You can see that it quickly purls as it starts to grow. It will keep growing, but now we move into the Perlin stable area. And so it will relax back on the Perlin, but it will keep growing because we're still in the meandering unstable area. And if we'll keep going with the simulation, then it will keep going, and it actually becomes negative. So it goes into the meandering stable area, and it will relax back into a ring. Another nice one last thing is that we have this system. So again, we're looking at bilayers and filaments. Only now, instead of just letting the system run, we change the tilt of the well, right? I'm using a tilted well potential. So we start with one bilayer and two filaments. You can see that in the second left, the filaments start to grow at the account of the bilayer, but quickly enough, the bilayer will purl and will create these bumps. So basically, we show that in these settings, the functionalized Cahn-Hilliard cannot support the coexistence of the two structures. Thank you.
Thank you, Noah. That is very interesting. Are there any questions?
May I ask one question?
Please, Vishnu, start.
Yeah, really interesting talk. The solution that you have presented, have you computed those solutions analytically, or did you use some numerical technique to compute those solutions?
The numerical simulations are numerically. I do only analysis. So I actually did it like slowly and painfully.
Okay, which techniques did you use? Finite difference, finite element?
What, to find in the numerics?
Yes.
Well, I can address you to the people who do the numerics. I do the analysis.
Okay, that's good.
So I'm more than happy to talk about how I separated the operator and projected it, but the numerics, I just love the pictures at the end.
Yeah. Anyway, thanks for the question, Vishnu. Did I understand it correctly? The epsilon is fixed, actually. Is it right?
Yes. Yes, we don't look at the gamma limit or something. Yes.
Yes.
We
Okay.
take it
So
fixed.
you fix the epsilon, and
Yes.
then you let t time to tend to infinity.
Exactly.
And then you check exactly what are, depending on the potential you get out. But it's interesting that it actually comes to such kind of perforation of the original area, so that you get holes back later then. This is very interesting to see. I would not expect, actually. My intuition is not, of course, I'm not in this theory, but it's from my superficial intuition, not clear. Yeah. Any other contribution or questions to Noah, please?
I just have a little thing. That's a beautiful talk, Noah. Thank you so much. I just want to check if I understand the situation correctly. So would it be correct to say that you have kind of some kind of a geometric object, two-dimensional, and you have a coupled mean curvature flow happening on vectors on the surface, and then a surface diffusion type flow of the object itself?
What do you mean by vector? So I do have so let's talk about spheres, right? So I have a sphere and it has its own normal velocity, right? And
yeah, that's the surface diffusion. That's the surface diffusion part. The sphere is evolving by
the meandering part, yes.
And then there's filaments, the
Yes.
spaghetti guys. I think of these as just vectors on the surface, but are they curved or are they just straight?
Oh no, they curve and they purl.
Oh.
So unlike the bilayer that curve and get these bumps, but the spaghetti ones, they curve and they basically break into tiny balls that have their tails pointing in. So it's actually it's codimension 3 already.
Yeah, that's so interesting. So how do you understand that filament flow? Is that a geometric flow? Like, can that be understood geometrically? It looked like a mean curvature flow, but by the equation, but I wasn't, obviously I didn't understand it.
Yeah, it's a mean curvature flow. So let me go back. Wait, okay. So if I go back, yep. So we have the bilayer and we have the filaments. And for the filaments, we do have a mean curvature flow. So we have a normal velocity that depends on the curvature. It is vector. So we are, if we go back to here, then when we look at the codimension 2, when we look at filaments, we do have a system. So now the velocity is in 2 directions. We have Z1 and Z2, and we have 2 normals to the surface. Yes.
Is this a degenerate, like a totally, like, is this a sub-Riemannian, sub-elliptic equation? Because you've got like also a second derivative of the curvature vector along the filament, right? But you don't have the.
It's like curve shortening plus a curve diffusion term with an epsilon coefficient, right?
Yeah, I'm just looking at the difference with the Z and the X terms, Z and the S terms.
Oh, so the S term here, it comes from the Laplace-Beltrami in this codimension 2.
Yeah, the Z's are orthogonal to the filament, or not orthogonal, but they're not on the filament, right?
The Z are the normal. The Z's are the scale distance from the interface itself.
It's such an interesting equation. All right. Anyway, I think the equation's really interesting. Okay. Sorry.
Yeah. I think I have to.
Yeah.
No, actually, we still have 5 minutes. I mean, Gia-Kun starts at 11. We can continue. Sorry, the discussion there. Now it's okay.
So anyway, I guess just to correct what I said at the beginning, I guess it's more like surface diffusion plus curve diffusion.
Well, I'm not sure. What do you mean by, so it's, I'm not sure.
But both equations are fourth order, right? Both equations are fourth order.
Yes.
Yeah.
Yeah, it's the same equation for both. Where is my? So it's the same equation for both. It just looks different for the codimension too.
And the coupling between them is by boundary condition essentially. So like as the surface moves, the base point of the filament moves, or is there like a more involved coupling?
So the thing is that there's a limited amount of lipids in the background, right? So to grow, they need to suck up the lipids, and to shrink, they need to release the lipids.
And is that background chemical potential, is that also evolving?
Yeah.
Oh,
no,
gosh.
no, well, the background is evolving because they're drinking it, right? So for example,
it is evolving.
we go back here. Oh, where's my nice video? So when we start, let it stop. When it stops, we have this nice symmetric form, and you can say, oh, is it always symmetric? Can you change it? And the answer is yes, we can do whichever shape you dream of, almost. I'm probably exaggerating, but the idea is that when we start in the system, and I tell you, we start with a bilayer and there's too much lipids in the background, the question is, where is the lipid, right? And you can see they go towards it. They want to drink it up, and they evolve towards the lipids in the background.
Mm-hmm.
Beautiful.
Mm-hmm.
It looks like that particular configuration has an initial rotational symmetry, which I suppose is preserved.
But no, that's the thing. It came out nice, and we kept this one. But we can do any figure that we want. So we can have 3. We can have an odd number of edges.
Mm-hmm.
Cool. Thank you, Noah.
Sure. Thank you very much, and thank you for inviting me to speak.